Welcome to Lecture 19, Regulation of Gene Expression. So, in the previous lectures, we've been talking about the mechanisms of gene expression, specifically how transcription is initiated, and then how mRNA is processed, and then finally how translation is initiated to regulate and generate the expression of proteins. But what we generally want to understand is how all of that stuff works together and to determine how the degree to which RNA proteins or products of gene expression are expressed and specifically regulated. So RNA levels or protein levels from a given gene can vary as a function of many different things. So that includes like the cell cycle, the time of day, the environment, the developmental stage, sex, so on and so forth. So RNA levels through transcription and protein levels through translation, those are fluctuating a lot. Sometimes genes are turned off and sometimes genes are turned on. So why would an organism modulate levels of the expression of either RNA or protein? One reason would be that RNA and proteins can be energetically costly. So it doesn't make sense to make RNA and proteins when they're not needed. So for example, if a cell isn't dividing, it wouldn't make sense for the cell to express the DNA polymerase at a very high level, for example. The other thing that's important to understand is that the expression of RNA and proteins with certain roles may be harmful at certain times. So, for example, genes that promote cell proliferation, might not be always great to express at high levels, because that can, for example, cause cancer. So if a gene is expressed at a very high rate, and the proteins are expressed at a very high rate, and the function of that gene is to, initiate multiple rounds of mitosis, then that could cause a tumor. So to understand how genes make sure that they are expressed at the correct time, it's important to understand that the regulation of gene expression can occur at many levels. So in this little diagram, we kind of have the whole story. We have DNA, we have transcription, the RNA is generated, the mRNA is processed, sent out into the nucleus. Of course, this is a eukaryotic gene expression that we're talking about, and translation occurs within the ribosome. And how the levels of RNA and the levels of protein are regulated can be determined at the transcriptional level. For example, how much transcription is happening. It can be occurring at the RNA processing level. Is the RNA processing efficient, and therefore is it able to exit the nucleus? It can occur at the translational level. For example, the control of translation, and we talked about, for example, that poly A tails can be important in regulating whether RNAs are translated or not. And then there's an important concept of mRNA stability. So you have an mRNA that is expressed and exits the nucleus. How long does that mRNA stick around? Does it stay at very high levels for a long period of time? If that's the case, then it might be producing lots of protein while it persists. However, it may be degraded very rapidly, and in that case, the expression of the protein might be, quite low, because while transcription could be high, there could be a lot of mRNA being produced, but the mRNA would turn over very rapidly, and so you could have a disconnect between the mRNA expression level and the protein expression level. In addition, after proteins are translated, there can be further modifications. We've actually talked a little bit about phosphorylation of proteins. And so protein modifications can play an important role, not in necessarily regulating the level of the protein, although certain protein modifications can target a protein for degradation, but they can also regulate the function of the proteins. So the pioneers of gene regulation did not work in eukaryotes. Francois Jacob and Jacques Monod from France in the early mid to early of the last period of the last century worked in E. coli. And in particular, what they were trying to understand is how E. coli metabolizes carbon sources, all right? And so we've talked a lot about that. We've talked a lot about how bacteria have systems in place that can break down certain carbon sources and make those carbon sources available for growth. And so in particular, the carbon source that they were looking for was lactose, because E. coli has an ability to break lactose down and metabolize it. And we've talked about lac mutans before already, and these are bacteria that have no ability to metabolize lactose. Okay, so let's remind you what lactose looks like. All right, it has these sort of two rings. All right, so it's a type of sugar, and there's a oxygen connection between these two carbon rings, and this is what lactose is. But there's an enzyme, and that enzyme is known as beta-galactosidase, and that enzyme has the ability to cleave this linkage between these two rings and break down lactose into galactose and glucose. Right? So when lactose is the only carbon source around, these are the products of breaking down lactose. It's important then you can see that the expression of beta-galactosidase is induced. So bacteria might find themselves in an environment where there's tons and tons and tons and tons of lactose, and perhaps that's the only carbon source. Clearly they need to develop a way to break it down. That can be initiated through beta-galactosidase expression. So that's the general principle, but what I want to actually zoom in a little bit, because I'm going to kind of use this terminology a few times, is that basically the bacteria have evolved a way of recognizing lactose in the environment and turning on the enzymes that allow lactose to be broken down. And by those enzymes, I mean beta-galactosidase, the enzyme that breaks them down. So there has to be a system in place for the bacteria to, number one, recognize that lactose is in the environment, and number two, in response to that lactose, upregulate or cause high levels of expression of beta-galactosidase. So basically, this is a sort of fundamental point about gene regulation. And this was a system that was dissected by Jacob and Monod. And the principles that they identified are the ones that I'm going to discuss next. These are the principles by which a simple bacteria has the ability to turn a gene on or off in response to, let's be honest, an environmental change. And in this particular case, the environmental change is the presence of lactose. So the principles that I'm going to illustrate are what they inferred through genetic analysis. And we'll touch on how they did genetic analysis to make these inferences, but I'm going to start off with basically their conclusions. Okay, so one mode of gene regulation, which is important for allowing bacteria or any organism, I suppose, but let's focus on bacteria here in E. coli, that allows them to recognize the presence of a breakdown product like lactose is shown with negative regulation. So what we have here is a gene in green, which is maybe being transcribed under some conditions or not. Now, negative regulation is mediated by a repressor protein, and a repressor protein binds to something called an operator. And when that occurs, the repressor protein blocks RNA polymerase. All right, we've talked about RNA polymerase already. And so when that blocks the access of RNA polymerase to the beginning of the gene near the promoter, it blocks transcription. However, if the repressor is absent, then we would not block transcription, we would have expression. This is a form of what is called negative regulation. And negative regulation is defined as the naked state of a binding site leads to transcription. So this is naked here. And this is important. The binding of a protein at the binding site leads to repression. So negative regulation is when protein bound to a regulatory sequence represses transcription. Now, there's another form of gene regulation which is called positive gene regulation, and this is not mediated through a repressor but is mediated through an activator. Now, when an activator is bound to a regulatory sequence, we'll call it an activator binding sequence, the presence of this activator drives transcription. All right? But the absence of it doesn't result in active transcription. So positive regulation we can define as the binding of the protein drive the expression. So in that sense, with respect to what the function of the bound protein is, it's opposite of negative regulation. And there can be a form of gene regulation called positive autoregulation, which means the production of a product stimulates itself. So you can imagine you get some sort of runaway circumstance, which could cause very high levels of expression, if the gene that's being transcribed in fact is activator. So we have activator driving the expression of a gene product, but if the gene product itself is activator, you're going to get more and more activator, and you would get basically almost a runaway process called positive autoregulation. Okay, so that's negative and positive regulation. In addition, there are regulatory effectors that are called allosteric effectors. And what allosteric effectors do is modulate the way that activators and repressors function. Here we have a case of positive regulation, but under certain circumstances there may not be a particular other protein or another molecule that mediates the positive regulation of the regulatory protein. However, under a case where an effector is present, all right, this is a particular molecule, what that molecule does is it changes the conformation of the activator, and now the activator is bound to it and we have positive regulation. Now in the case of negative regulation, we have a repressor that would be bound always to the operator, all right, but then a allosteric effector binds to the repressor protein and pulls it off. So we still call this one positive regulation because when the protein is bound to the regulatory sequence, it drives transcription. And we still call this negative regulation because when the protein is bound to the regulatory sequence, it stops transcription. All right. But the circumstances by how the effector works clearly are different in these particular cases, because here the effector allows the regulatory protein to bind, but here the effector prevents binding. Okay, so going to negative regulation, okay, and so this is going to be, here's an inducer, this is going to be an allosteric effector. All right, we're going to show a form of negative regulation which is regulated through an inducer molecule, an allosteric effector known as an inducer. So here we have a repressor protein that's blocking transcription, but then we add this inducer molecule. It interacts with the repressor falls off, right, and we drive transcription. And this is called inducible transcription because an inducer pulls a repressor protein off. Okay, let's think about a circumstance why this type of gene regulatory network might be helpful. If an inducer is a product that needs to be broken down, say for example lactose, right? If an inducer is a product that needs to be broken down, this system can increase production of necessary enzymes in presence of the product. So this inducer might be a protein, might be some sort of product that needs to be broken down. Well, then by the inducer binding to the repressor and driving transcription, by the repressor falling off, the transcription of genes from this particular region could lead to the breakdown of the inducer, which might be beneficial. Now you might say to yourself, well, why do we call this negative regulation? Because we have the inducer, it pulls off the repressor, and we have the increase of transcription here. Let's be clear, the reason we call this negative regulation is because when the repressor is bound to the regulatory sequence, transcription is blocked. Now the circumstances by which whether the repressor is bound or not clearly can depend on these inducers, all right? But the point here is it's negative regulation because when the repressor is bound, you block transcription. When the repressor is off, transcription is enabled. Okay, so now we'll continue an alternate version of negative regulation. Here in this particular case, we have something called an aporepressor, and the reason it's called an aporepressor is it doesn't bind in its naked state. But in conjunction with a corepressor, all right, these two come in contact with each other, and then they bind the sequence, and here they block transcription. Now this is still negative regulation because when the regulatory sequence shown in yellow is bound by protein, it prevents transcription. All right, but the conditions obviously are different than the previous case because here the aporepressor binds to the regulatory sequence in the presence of this particular co-repressor. All right, so this is called repressible transcription because a co-repressor assists in shutting down production. When would this be helpful? If a co-repressor is a product that is in excess, all right, so it's some sort of, maybe something involved in amino acid synthesis, for example. It's like, oh, there's too much arginine here, all right. If a co-repressor is a product in excess, it can shut down production of proteins that lead to production. So, whoa, there's way too much arginine in this scenario. Okay, well, we are going to turn off the genes now that are involved in synthesizing arginine. Okay, again, negative regulation. Too much of me. Okay, so this gets to this question which we were sort of thinking about is, how does E. coli, quote, know, all right, not cognitively, of course, but there's a system in place that recognizes when lactose is in the environment and beta-galactosidase needs to be produced. So how does this system of regulation recognize the presence of lactose being the perhaps sole carbon source? Okay, well, that's the key question here, but there's one more thing I want to talk about before I give you the answer for that. All right, so the lac operon is the gene, basically, that was used to dissect this fundamental question of how an organism recognizes something in its environment and adjusts gene expression in accordance. All right, and the lac operon expresses not just beta-galactosidase, it also expresses two other proteins. One of those is called permease, which pumps lactose into the cell. All right, so when there's lactose in the environment, and you want to pump it in so that you can use the lactose. And then there's another gene called the A gene, or also known as the transacetylase. It's not critical for lactose catabolism, so we're not going to get into the weeds on that. But the point I want to make here is that this is something called polycistronic mRNA. All right, so here we have a promoter and operator. These are the regulatory sequences, and a single mRNA is transcribed by RNA polymerase. Now, in eukaryotes, this would not be this wouldn't be happening, but in prokaryotes, a single mRNA can encode for multiple proteins: protein 1, protein 2, and protein 3. Now, how does this happen? What this means is that this mRNA, in some cases, has to be translated start codon to stop codon here. In other cases, needs to be translated start codon to stop codon there. In other cases, needs to be translated start codon to stop codon there. So how does, you can kind of see, well, this first one makes sense. Start to stop, that can work. How does translation initiate off of a single mRNA molecule to produce the permease and the transacetylase? Well, the key here is the way that translation works in prokaryotes. Remember the Shine-Dalgarno sequence? If there's a Shine-Dalgarno sequence in an mRNA that is preceding methionine, that can be translated. All right. So likewise, if there's a product here like a Shine-Dalgarno sequence and then a start codon here, you can get an internal translation site and then another internal translation site. Now, of course, this does not work with eukaryotes because there's a cap and the first methionine is scanned for, and once that first methionine is identified, translation starts there, but there's no internal translation that can occur because the scanning begins from the cap. Now, this is the structure of the gene that is known as the lac operon, and I would like to just briefly mention that these two genes, which are identified to be important for the metabolism of lactose, the beta-galactosidase gene known as Z and the permease gene known as Y, those were identified to be nearby each other, and specifically, eventually found to be, part of the same gene. That was found by mapping with co-transduction. Remember, we talked about co-transduction mapping can be used to identify whether genes are nearby other genes. Well, in this particular case, co-transduction experiments indicated that LAC-Z and LAC-Y were right nearby each other. Okay, so how does this work? How does the lac operon work? Well, if you take bacteria, E. coli in particular, and they're not on carbon source, they're not on a lactose carbon source, if you add lactose to the media, what you'll see is the lac mRNA takes off like a rocket. Okay, but you can add lactose, but you can also remove lactose. And the moment you remove lactose, you'll see that the mRNA starts to go down. All right, so what's happening here is you add lactose, transcription is initiated at great level. You remove lactose, and now transcription basically stops. So where does all that extra RNA go? The excess RNA is broken down. Now, that's the blue line showing the mRNA, but there's a delay with respect to the protein products. Here we're just talking about beta-galactosidase and the permease. You can see that there's a bit of a delay. And why is that? Well, it takes time for translation to occur. So the mRNA can reach high levels, but then it takes a little bit of time for the mRNA to be translated. So there's a bit of a delay. So the lactose is added, the mRNA shoots off like a rocket, and then a little bit later translation occurs, and beta-galactosidase and permease are expressed. Here they're expressed at low levels, and that is just basically determined by how much translation is occurring. But what's interesting is even after lactose is removed, the beta-galactosidase and the permease persist. And so what that means is that the protein products stick around, but the mRNA does not. So the way that the lac operon works, there's fast induction when lactose is added to media, and there's fast downregulation of the mRNA when lactose is removed. So how does it do this? Well, you might imagine one form of regulation that occurs is through the recognition of the lactose itself. All right, and so there's a protein that's also encoded. So I talked about Z, Y, and A. There's also a protein called LAC-I, which produces a repressor protein. So LAC-I is transcribed, and the mRNA is generated and translated here, and you get a repressor protein. The repressor protein binds to the operator and prevents transcription when there is no lactose. So this is clearly negative regulation. When there's no lactose, there's no beta-gal because of the negative regulation. Okay, but lactose, or chemical analogs of lactose, can act as the inducer. And the inducers bind to the repressor and it falls off the operator. So here we have the repressor molecule encoded by LAC-I, mRNA, you get the translated product. But when this inducer protein, or chemical analog, in this particular case lactose itself, comes in contact with the repressor falls off. It can't bind to the operator, and transcription ensues and leads to the production of beta-gal. Okay, and that inducer itself is lactose. So lactose itself, as a chemical, regulates the expression of the lac operon. So then without the repressor binding to the operator, transcription ensues. And so here's the RNA polymerase. Transcription is initiated there. You get the transcription of the lac operon, you get the lac mRNA, and then you get downstream translation of the beta-galactosidase, the permease, and the transacetylase. And then that beta-galactosidase, of course, can break down the lactose. So we'll go through this extensively in class on Tuesday, but what I wanted to sort of provide an introduction of is how the genetic analysis of the lac operon revealed these principles of negative regulation. All right, and the critical point is that the geneticists that were analyzing this system of gene regulation considered inducible to be the wild type phenotype. So they investigated that phenotype. And by inducible, what I mean is you add lactose to the media, you express beta-galactosidase, you remove lactose from the media, you no longer drive the transcription of the lac operon. Okay, so that's the wild type. You can turn on beta-galactosidase production when lac is added and then turn it off when it's removed. So there are different types of mutants, right? Some types of mutants of the lac operon are called constitutive, meaning they're always on. So lactose is in the media or not, doesn't matter. The lac operon's, expressing gangbusters. Then alternately, you can also have another type of mutant called uninducible, and this is always off. So add lactose, remove lactose, doesn't matter. The expression of beta-gal is completely off. All right. So just to be clear, by off and on, what we mean is whether the lactose can be metabolized by beta-gal. So you could say, well, it's like at the RNA level, it's transcription, but these were geneticists, and what they were trying to analyze was, bacterial genetics in the 1940s. So they weren't measuring RNA levels, but they were just using an assay that says, can lactose be metabolized by beta-galactosidase or not? Another interesting thing is that genetic analysis can be performed using what are called partial diploids. We've already talked about the F plasmid. All right, the F plasmid has a capacity to move bacterial genome sequence from one bacteria to the other. Well, one of the types of genes that can land on the F plasmid that can be used for genetic purposes are F prime lac plasmids. So these are plasmids that actually have the lac operon on them. And you can produce different versions in different genotypes. So you can have F prime lac minus, so this is a F plasmid that is defective for the lac operon. And you can have a lac plus bacterial genome. Or you could have F prime lac plus, so you'd have the fully functioning lac operon on the F plasmid, but you could put that into a background that is lac minus. So these are called partial diploids because you don't have the full bacterial genome in 2 copies, but you have a part of it in 2 copies, and specifically you have the lac operon in 2 different copies. So in order to understand genetic analysis of the lac operon with respect to negative regulation, one point is that protein products of the lac operon are diffusible, so they can rescue defects or complement another operon, those defects in another operon in those partial diploids. But regulatory sequences, and oftentimes we mean the operator, are what is called cis-dominant. So they strictly control the operon that they belong to. And we'll talk about that extensively in class on Tuesday. Some alleles of the lac operon that are going to be important to understand. One is the LAC-O-C, constitutive operator. So the constitutive operator, the repressor can't bind to it, and so it's cis-dominant. So it's always driving expression of that particular lac operon. All right. This is the deletion of the repressor. It's a deletion of the operator. So then there's also LAC-I. LAC repressor is the LAC-I protein. S, all right, which is super repressor, means that the repressor cannot be removed, so it's permanently stuck to the operator. LAC-P minus, so this is uninducible defective promoter, so the polymerase can't bind. So this is also cis-dominant, so it regulates the lac operon that it's joined to, but it doesn't regulate other lac operons that might be present on an F plasmid. And then LAC-I minus means that there's no repressor, a defective repressor, but it can be rescued by, this F prime lac plus plasmid. All right, and then other types of mutants are LAC-Z minus or LAC-Y minus, and those are basically defects in the proteins themselves. It could be some sort of deletion or stop codon that's premature inside the genes for beta-galactosidase and permease. Okay. So going back to this question of how does E. coli know when to make beta-galactosidase, that's how it works. So when lactose is present in the media, it pulls the repressor off and it drives the expression of beta-galactosidase. But that's not the only possible carbon source. So in fact, lactose is a complex carbon source, but galactose and glucose are much simpler carbon sources. So what this means is that when bacteria have a rich source of lactose and a rich source of glucose, it doesn't make sense for beta-galactosidase to be expressed to break down the lactose to glucose because there's plenty of glucose already there. So this is an interesting situation. You might say, oh, there's lactose in the media, so you should express beta-galactosidase, but there's also glucose in the media. So how do you prevent the expression of beta-galactosidase from being very high? Because there's no purpose in expressing beta-galactosidase because lactose is only broken down to glucose anyway. And if there's plenty of glucose, then what's the point? Okay, so there's going to be another form of regulation that will actually regulate the lac operon. and I'll walk through this. 1. When glucose is absent from the media, a compound known as cyclic AMP is at very high levels. It's kind of like a stress kind of sensor. All right, that's the simplest way to describe it. And glucose is very low levels. Well, then that means the bacteria is probably under a significant amount of stress because there's no carbon source. So cyclic AMP is at high levels. Okay, when cyclic AMP is at high levels, a protein complex, which includes CAMP or cyclic AMP, and another protein called CRP, what those do is they have the ability to activate transcription of the lac operon. All right, so that complex can bind to the promoter and drive transcription. So what this means is that low levels of glucose lead to high levels of cyclic AMP, and that leads to high levels of the lac operon activity. And so I'll show you how this works. So here's the CAMP-CRP complex. It is going to be with very high levels of CAMP. A lot of this complex will be bound to the promoter. And this will occur when there's low levels of glucose because there's an abundant amount of cyclic AMP, and that will drive transcription. However, when there's high levels of glucose, this positive regulator is not in great abundance. There's low levels of it, and that means you won't have very high levels of transcription. All right, So what I've just described to you is a form of regulation that's not negative regulation, but is in fact positive regulation. All right, so the positive regulation of the CAMP-CRP complex drives transcription, but there's also negative regulation. So let's talk about the conditions and how it actually synthesizes high levels of transcription of the lac operon. So condition 1, there's a lot of glucose and there's no lactose. Okay, clearly under that condition it makes no sense to express beta-galactosidase. And so because there is no lactose, the repressor will be bound to the operator. That would block transcription. In addition, we don't have any positive regulation driving transcription because the CAMP product is low, and so the positive regulator is not highly abundant. Now, you can imagine that you could, even though there's no lactose, remove glucose from the media. Now, this would cause high levels of CAMP, inducing basically a stress response. But there's no lactose, so even though the cell is stressed, there's no sense in producing beta-galactosidase. Okay, so in that particular case, the repressor is still bound blocking transcription, but this positive regulator is bound to the promoter, but there's no effect. All right, now let's do a thing where we add lactose to the media. So we have no glucose in the media. You add lactose to the media. Whammo! The repressor protein binds to the inducer, in this particular case lactose. It falls off, and then all that remains is the positive regulation. All right, so this CAMP-CRP complex now drives a high level of transcription because the negative regulation has stopped in that case. Then you can add glucose to the media. Okay, again, it makes no sense to express beta-galactosidase in that particular case. The CAMP levels go down again. You'll still have the lac operon no longer binding to the repressor protein, but the positive regulation is no longer active at the promoter, and so here we get no transcription. So the circumstance in which you get high levels of transcription is when there's lactose in the media but no glucose. Now that is prokaryotic gene expression, the principles of negative and positive regulation based on, the environment that the bacteria are experiencing. Obviously, there are many other ways in which gene regulation occurs in bacteria besides those, but those are the key takeaways. Okay, but I'm going to be a little dismissive here. I'm going to sort of argue that bacteria, while it's interesting to understand gene regulation in gene expression in bacteria, they are simply sort of bags of metabolism, right? So, they can sense the environment and turn genes on and turn genes off, but they don't have something that a lot of eukaryotes do. Now, not all eukaryotes have this, but they don't have something that a lot have, which is what I would call a highly complex development. All right, and by highly complex development, really what I mean is, starting from, just a few cells and, determining like, what are the circumstances, what are the processes that happen within all these cells that lead to either the production of this, this really cute baby or this like nasty warthog. Like what happens from here that causes either a cute baby or a nasty warthog? So, that's a big question. Like, and then a miracle happens, right? So this is a big, interesting thing that people want to understand in the field of biology. So I would argue that fundamentally, this point about development is basically the production of the adult form and all of the steps that happen. I would argue that development is fundamentally a challenge in gene expression. Specifically, how are genes regulated to allow a single cell to become a complex organism with 220 different cell types? And there's probably more cell types than that. Just as a very simple case, we'll talk about hematopoiesis, which is the development of blood cells. There's a hematopoietic stem cell, all right, and then that stem cell can either differentiate into a common myeloid progenitor or a lymphoid progenitor, and then further differentiation can produce all of these different types of cells. Now, obviously, the red blood cells are critical for carrying oxygen through the blood, but all of these other cells have highly specialized and different functions in the immune system. And how all of these different cells are generated from a common sort of cell through a lineage is actually what we would call development, and it occurs by certain genes being turned off and on here, and then certain genes being turned on and off here and on and off here. And it's sort of like how genes are turned on and off determines how this lineage of differentiation occurs. So what we're going to walk through briefly now is some big differences between gene expression and the regulation of gene expression between prokaryotes and eukaryotes. We've already talked about a few of them. Number one, eukaryotes have nucleosomes that add a layer of complexity to transcription. All right. So remember we talked about the 30 nanometer fiber. The nucleosomes are going to actually interact with the machinery of transcription. Prokaryotes don't have nucleosomes. So because of that, you could argue that basically bacterial DNA is, quote, naked, meaning it's not sort of bound up in a bunch of proteins. So what this means is that naked DNA in bacteria favors activation as the default state. And by default state is basically what I'm saying is a state in which RNA polymerase is bound to promoters and then drives expression. So in bacteria, activator and RNA pol, all right, we have naked DNA. The ground state is on, all right? But in eukaryotes, because the nucleosomes are compacted and there's all these regulatory sequences embedded in these DNA sequences that are wrapped around the nucleosome, in order for transcription to occur in that context, RNA pol2 has to be bound to the regulatory sequences. But they're embedded within this chromatin. So the ground state in eukaryotes is off. Now, certain circumstances can happen, and so repressor proteins can bind to regulatory sequences in bacteria, and that can turn gene expression off. So here we have a repressed state, which is off. But a whole bunch of different things have to happen in order for activation to occur in eukaryotes. Specifically, all of the nucleosomes need to sort of be unwound to gain access to the regulatory sequences, and then there's a whole bunch of transcription factors and regulatory sequences that drive transcription. So the key point here is that in bacteria the ground state is on, but in eukaryotes the ground state is off. So that being said, eukaryotes of course have to turn on assemblages of genes, keeping in mind that eukaryotes don't have these polycistrons, right? Like the lac operon, in which the transcription of a single mRNA can lead to multiple protein products. So because of that, gene regulation occurs differently in eukaryotes. Okay, So there are two kinds of DNA sequences in eukaryotes that promote transcription. Of course, there are promoters. Those are the sequences in the DNA that bind the transcription machinery. They often contain a TATA box. They're located right next to the gene, and they bind RNA polymerase to initiate transcription. But there are other sequences in eukaryotes that are called enhancers. These are often called upstream activation sequences or UAS sequences. They can be far away from the gene. And what they do is they bind proteins that then in turn recruit RNA polymerase. So you have these two regulatory sequences. This is going to be important for determining how a bunch of genes can be turned on or off at the same time. And the way that that works is basically that proteins can act by binding enhancers across multiple genes. So here we have a regulatory protein, all right? And these little bubbles are regulatory protein products. But under certain circumstances in which this gene is turned on, they can bind enhancers here, 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 and here, and all drive transcription. Why would it work like this? Why not just drive transcription of a single gene? Well, remember what I said before. A single transcript in eukaryotes, for the most part, only produces one protein product, in contrast to what happens in prokaryotes. In eukaryotes, you just get one protein product. But because of the Shine-Dalgarno sequence and the way that translation is initiated, this can all occur in prokaryotes at a single gene. But now, in order for it to work in eukaryotes, it has to happen across multiple genes. So, these regulatory proteins can recognize DNA sequences and bind and interact with other DNA binding proteins, and they influence that chromatin compaction and then act as a sensor or signal to other proteins. The point here is that under some circumstance in which this green gene product is expressed at a high level, it can turn on a whole assemblage of genes. This can allow coordinated regulation of multiple genes under the right conditions. And the system that we'll actually discuss for thinking about how this works is using, again, a carbon source mutation, carbon source gene cascade, but instead of in E. coli, we'll do this in yeast. So this is a biosynthetic pathway. The book goes into a little bit more detail. Not a biosynthetic pathway, a metabolic pathway in which galactose is broken down through many genes, GAL2, GAL1, GAL7, GAL10, GAL17, broken down to allow for glycolysis. And this large set of genes, 1, 2, 3, 4, 5, here in this particular case, all of these genes need to be expressed when galactose is present. All right? It's not an operon, but it's a co-regulated set of genes. So how does that occur? All right? Well, the critical protein that lies at the heart of this is called GAL4. GAL4 binds very tightly to UAS, or upstream activating sequences, and what it does is it has two parts. It has a DNA binding motif and an activating domain, and by binding to upstream activating sequences above this set of genes, GAL7, GAL10, GAL1, GAL2, it will recruit the transcriptional machinery in the presence of galactose. So that's the question, is like, how does it do that under the right circumstances? So this protein will bind these sequences and recruit RNA polymerase. Okay, so we know that it occurs through this sort of two-part system is because there's this activation domain and this DNA binding domain. You can generate mutes in which you cleave off the activation domain. So under normal conditions, you can have a reporter, say for example LAC-Z, you can use that as a reporter. Under this particular case, when this GAL4 complex binds to GAL4 activating sequences, it can drive transcription. But if you cleave off the activation domain, you don't have the expression of LAC-Z. So this is a two-part system. Part 1, binding the DNA. Part 2, the activation domain, which recruits the RNA polymerase. Okay, so how does the sensing work? In the absence of galactose, there's another protein called GAL80, which binds to GAL4. All right, so here we have in the cell upstream activating sequence, and we have a GAL gene here, and GAL4 is bound to it, but GAL80 is bound to, I'll go back, the activation domain. And because GAL80 is bound to the activation domain, GAL4 cannot pull RNA polymerase and drive expression. Now, there's another critical protein involved in the regulation of all of these different GAL genes, and that's called GAL3. And it hangs out in the cytoplasm. This GAL3 is the sensor of galactose. When galactose is present, all right, there's a conformational change through the interaction with GAL3. And when GAL3 changes its configuration because it's bound to galactose, it actually recruits GAL80 away from GAL4, and the GAL80 is sequestered in this complex. So in the presence of galactose, the GAL80 inhibitor is sequestered. Now we have GAL4 is no longer blocked, and it can now recruit this transcription complex and initiate transcription. One final point I'd like to make here is that the regulation of genes through these proteins that bind DNA is an extremely important feature of gene regulation. And there are some very many different kinds of proteins that do this, but one of the most famous ones are proteins that are known as zinc finger proteins. And GAL4 is an example of a well-studied group of proteins that bind DNA that have this two zinc ions that are function within the protein complex. And here's a three-dimensional structure. Here you have zinc ions in yellow. Here you have zinc ions in yellow. And GAL4 acts as what is called a dimer. It acts in concert. There's two copies of GAL4. And the way that these zinc fingers interact with the DNA is based on the is based on the DNA sequence of the DNA itself. And so particular zinc fingers will have particular affinities for particular DNA sequences. And this is what enables, when conjugated with an activating domain, DNA sequences to play a critical role in the regulation of genes.